Hi everyone, this is week 3 out of 4 in our topic of carbohydrates. And today we're going to cover two main topics. The first is glycogen, and the second is molecules that are complex conjugates of carbohydrate and protein that have specialized functions in the body. So our first topic is glycogen metabolism and glycogen storage diseases. Our learning objectives are to list and discuss glycogen synthesis reactions as well as glycogen degradation reactions. We also want to explain the regulation of glycogen synthase and glycogen phosphorylase and discuss glycogen storage diseases. So glycogen is our primary storage form of glucose. It's found mainly in the liver and the muscles, though the kidneys and intestines are also minor storage sites of glycogen. Now, the glycogen is a readily available source of glucose to supply the tissues with an energy source. However, that really refers to the glycogen that is present in the liver. So skeletal muscles also store glycogen, but they're lacking the enzyme glucose 6 phosphatase. So they're not able to really convert the glucose that's locked up in a glycogen into a free glucose that's available for other tissues. Rather, the muscle needs to utilize the energy of glucose 6P directly. And we're going to talk a lot more about that. Okay, so our major site of daily glucose consumption is in our brain, or I should say glucose consumption via aerobic pathways is in the brain. So out of the average 160 grams of glucose that we consume in a day, 120 grams or 75% is used for brain function. And most of the remainder is used for our red blood cells, skeletal muscle, and heart muscles. Now the body can either obtain glucose directly from the diet or can produce glucose from glucose neogenesis. And that can occur either from lactate or from amino acids. And glycogen allows glucose to remain soluble in polymeric form in our body fluids. Now, the liver has the highest specific content of glycogen of any body tissue. So, and glycogen is 10% of the liver's weight in glycogen. Now, the muscle tissue has a much lower concentration of glycogen per unit mass, but because we have so much more muscle tissue than liver tissue, we actually have twice the amount of glycogen stored in our muscles compared to our liver. But since our muscles are lacking the G6P phosphatase, only our liver glycogen is considered the major buffer for blood glucose levels because only our liver glycogen can release free glucose to impact our blood glucose levels. So next we're going to talk about how glycogen is formed, which is a process known as glycogenesis. Okay, so this occurs when a glycogen polymer is in a linear array of, let me back up. The glycogen polymer is a linear array of glucose residues with alpha-1,4 glycosidic bonds. So you can see that here in this graphic. So we have a linear glucose molecule with alpha-1,4 bonds. However, the glycogen molecule also has branch points that are alpha-1,6 glycosidic bonds, and then those occur every 8 to 10 glucose residues and allow for this extensive branching of the glucose molecule. And why do we want this extensive branching? Well, it allows for really an ability to build these glycogen molecules really fast because we can add on this glucose molecules to all these different branch points, and then it also allows for really quick degradation of the glycogen molecule in order to allow us to have quick access to free glucose in order to meet our needs. So I'm going to walk through every reaction of glycogen synthesis. And in these reactions, please keep in mind that we're talking about the addition of the glucose onto the glycogen molecule via these alpha-1,4 bonds. And we'll talk about branching next. So the first reaction of glycogenesis is to convert glucose that we get either from our diet or gluconeogenesis to G6P. And you'll note that this is the same reaction, first reaction as glycolysis, which we're going to talk about next week. So if we're going to oxidize glucose for energy, or if we're going to add it onto glycogen, the first step is the same. Now, the enzyme for this reaction is glucokinase in the liver, but hexokinase in the muscle. So glucokinase in the liver has a lower affinity for glucose. So this enzyme is only going to be active when glucose levels are high. So high glucose levels in the blood are a signal for glycogenesis. And this makes sense. We've got lots of glucose. We want to build it up and store it via using glycogen. Now, hexokinase has a higher affinity for glucose. So this means even when glucose levels aren't that high, the hexokinase enzyme can work to build glycogen in the muscle. And this is because other tissues need glucose, even if we don't have high concentrations of it. They still need to utilize it. So in the muscle, G6P inhibits hexokinase. So this really makes sense. We have a reaction that's converting glucose to glucose 6P, and our product is going to inhibit our reaction. So that makes sense. If the muscle's not using up the G6P, then we can stop making it because we have more than enough. However, this is not the same type of regulation in the liver. So G6P does not inhibit glucokinase. So even when we have a conversion of glucose to G6P, we're not inhibiting that reaction. And the idea behind this is that it allows the liver to remain active in storing as much glucose as possible in the presence of high glucose levels. So it's not activated until, we'll remember that this enzyme, glucokinase, is not activated until we have high glucose levels, but then it's going to keep working and working and working to store as much of that glucose as possible into the glycogen molecule. So our next reaction is the conversion of G6P to G1P via the enzyme phosphoglucomutase. So we're really just moving the phosphorus from one carbon to another on the glucose molecule. And then reaction 3 is our energy requiring step, where G1P in the presence of UTP is converted to inorganic phosphate, and then the addition of the UDP onto glucose. So UTP, you can just think of as kind of similar to ATP. It just has the different base, the U instead of the A. So we're using up UTP in this reaction. It's energy requiring, and we're adding the UDP onto the glucose. Okay. And then in our last reaction for adding these glucoses onto the glycogen polymer with the alpha-1,4 bonds is that we have our glucose polymer that's already started. We add on the UDP glucose, and with the enzyme glycogen synthase, this is an important one, we're able to add the glucose onto this original glucose polymer, and the extra UDP is released in free form to be resynthesized to UTP, presumably. So the glycogen synthase utilizes the UDP glucose as one substrate and the non-reducing end of the glycogen as the other substrate in order to add the glucose onto the glycogen polymer. However, for each new glycogen molecule, a primer is required for attachment to the first glucose molecule. So first, we really need this primed molecule in order for glycogen synthase to be able to add these glucose molecules on via the alpha-1,4 bonds. Okay, so as I just said, the primer is required for attachment of the first glucose molecule onto the new glycogen molecule. And this primer is called glycogenin. And this is a protein that we have found is located at the core of glycogen molecules. And this protein actually catalyzes its own glycosylation, or the addition of glucose onto it. So glucose attaches the glucose residue of UDP glucose to the hydroxyl side chain of a protein residue. And I've seen different proteins talked about for this reaction, either a serine or a tyrosine. But the important thing to remember is that it's a protein residue um, that catalyzes its own glycosylation and therefore is able to add these glucose molecules on to form this kind of primer for the glycogen molecule. Now, when the chain is long enough, it serves as a primer for the glycogen synthase to come in and elongate the polymer by adding on the glucose. So glucose synthase catalyzes the addition of the glucose moiety in the alpha-1,4 orientation, but again is not involved in the branching of the glycogen molecule. So here's just a diagram that demonstrates what we just talked about. So we have the glycogen primer, which involves a protein residue, and then we have this primer is self-glycosylating, so it's able to add on 8 glucose molecules. And then we have this protein residue with 8 glucose molecules, which is termed the primed glycogenin. And this primed glycogenin serves as a substrate for glycogen synthase to add on molecules in the alpha-1,4 bonds, so the linear and also the branching enzyme, which we're going to talk about, to make the branches. And therefore, what we end up with is a glycogen complex. Okay, so we have the beginning of a glycogen molecule, which is a linear polymer, but how do we get these branches on it? Well, we do this with the branching enzyme, also referred to as amylo-4,6 transferase, but usually branching enzyme. Now, the branching enzyme catalyzes the transfer of the oligosaccharide chains. So remember, oligosaccharides are chains of about 7 glucose residues. And so the branching enzyme catalyzes the transfer of these shorter glucose chains from the non-reducing end of the linear glycogen segment and transfers them on to a interior glucose using the alpha-1,6 bond. Then, once this branch is added on, both branches can elongate with glycogen synthase until they're long enough to produce new branches. Now, as I said, branching increases sites for synthesis and degradation in order to enhance molecule solubility and our access to glucose when we need it. Okay, and this is just a diagram showing what I just talked about. So we have our glycogen primer here, and then we have the addition via 1,4 bonds, and then we have our glycogen branching enzymes that comes and cleaves off this non-reducing end of the glucose polymer, and then adds it on to the an interior glucose with an alpha-1,6 bond. And then the glycogen synthase can come along and keep adding on glucoses in the 1,4 until again they're long enough in which the branching enzyme comes and makes another branch. So here's just kind of an overview of what we've talked about with glycogenesis. So we have our glucose. Let's say they're in really high concentrations, so we want to add them on as storage and store them. So here we have, sorry, I'm trying to get my arrow. Glucose. Then we have the addition of a phosphate through ATP using either the glucokinase enzyme in the liver or hexokinase in the muscle. If it's in the muscle, then the G6P is just converted to pyruvate and then oxidized for energy. However, if we're in the liver, the G6P can be converted to G1P, then to UDP glucose. The UDP glucose is added onto the glycogen primer in order to grow the linear glycogen molecule. And then through the use of utilization of the branching enzyme, branches are added onto the glycogen molecule and it grows. Okay, so we know that glycogen synthase is our really important enzyme for increasing the glycogen size, but how is this enzyme regulated? Well, it's present in 2 forms, the active form A an inactive form B. And it's the phosphorylation of the form A to the form B that inactivates the enzyme. So various protein so again, let's think back to the beginning of the course when we talked about kinases, which add a phosphate group onto a certain substrate. Okay, so various protein kinases are activated by different intercellular signal transduction pathways, such as a hormone or a nutrient signal. So the hormone or nutrient signal is the first messenger, and then activates protein kinase, which phosphorylates glycogen synthase. So, which makes it inactive. So we're going to go through this a little bit more, and I'll explain it in a different way. Okay, so we know again that we have hormone signals or nutrient signals. These are the first messengers. They let's see here, and then they signal protein kinase to phosphorylate glycogen synthase. Okay, and here's just kind of a list of the protein kinases that can phosphorylate glycogen synthase. So when we have the active form of glycogen synthase, a protein kinase can add a phosphorus group, and then it makes it inactive. So intracellular CAMP concentrations activate protein kinase A, which regulate glycogen synthase and glycogen phosphorylation. Okay, so when I was saying we have our hormone or nutrient signals, which are first messengers, these activate increased CAMP concentrations, which are second messengers, and these activate protein kinase A, which phosphorylates glycogen synthase and therefore inactivates it. Okay, so we can also go the opposite way, where we can have inactive glycogen synthase, and then we can take away the phosphorus group that made it inactive through the use of an enzyme, which is a phosphatase. And then when we take the phosphate away, then again, we have the active form of glycogen synthase. So just think, active, add a phosphorus, then it becomes inactive. When we take away the phosphorus, it becomes inactive. So the enzyme phosphoprotein phosphatase is regulated by an inhibitor 1 in certain tissues, and especially in skeletal muscles. So if we think about this, this makes sense. So we have the inactive form, B, and we want to make it active in the muscles. And this makes sense because these have a high affinity for glucose, but because the muscles need the glucose even when our glucose levels aren't high. So when we have our hormone signals such as glucagon or epinephrine, it activates CAMP synthesis, and then CAMP, which is our second messenger, remember, activates protein kinase, which phosphorylates or inactivates glycogen synthase. So the hormone just depends on the location. It's glucagon in the hepatocyte receptors. So just kind of remember back to your past classes, when glucose levels are high, when glucose levels are low, we have the hormone glucagon. So, and we see here that glucagon inhibits glycogen synthase. So that makes sense because if we have low glucose concentrations, we don't want to store the glucose we have. We want to have it be available for our energy. And same thing, epinephrine in the muscle cell receptors. So we think of epinephrine as a stress hormone. And when we have a stress hormone, we don't want to store up our glucose. Rather, we're kind of catabolizing and using it up. Okay, so here is just a graphic. I'm just going to talk about the top part of this. We'll come back to this graphic in a bit. But here we have the epinephrine and glucagon hormones. These activate CAMP, which activates protein kinase A. The kinase is responsible for adding the phosphorus group onto the active form of glycogen synthase to inactivate it. So basically, if we have these stress hormones saying there's not enough glucose or we need to have catabolism, then that's going make the glycogen synthase inactive. We don't want to store when we need these, when we have these hormones present. But conversely, if we have the protein, conversely, if this isn't present, this protein kinase isn't present, or if we have insulin, it's going to have a negative effect on CAMP, negative effect on protein kinase A, and glycogen synthase is going to stay active so that we store the glucose that we have present. Okay, so now we've talked about glycogenesis. Now we're going to talk about glycogenolysis, which is glycogen degradation. Now, the major enzyme in glycogenesis is glycogen phosphorylase, which phosphorytically removes a single glucose residue from the alpha-1,4 linkage in the glucose polymer to produce G1P. And again, we're going to go through these reactions in a step by step. Okay, so here we have our glucose polymer from our glycogen. And then we have the addition of an inorganic phosphate. And this addition of the inorganic phosphate allows a glucose to cleave off of the glycogen. So therefore, we have a glycogen with one less glucose and then a glucose that's phosphorylated. And this reaction is repeated several times to remove the glucoses from the linear glucose polymer until we reach a branch point. Now, when we reach a branch point, we utilize a debranching enzyme. And there's two parts to the debranching enzyme. So the debranching enzyme has a transferase. So the transferase is an enzyme that catalyzes the transfer of 3 or 4 glucose units closest to the non-reducing end of the glycogen and attaches them to the end of a longer chain with 1,4 linkages, okay? So just think we have a glycogen molecule that's linear with a branch point, okay? So we have the transferase enzyme, which comes and takes some of the glucoses on the branch point and then adds them to the long glucose linear polymer with a 1,4 bond. And then we have the alpha-1,6 glucosidase, which hydrolyzes the remaining glucose at the alpha-1,6 branch point and releases it as free glucose. And I have some diagrams here, which I think will make this a lot clearer. So, okay, so I think that, let's see, is that on the next slide? The graphics in a couple more slides, but we'll get to that. So after this reaction 2 and 3, we then have reaction 4. So we have our G1P, our phosphorylated glucose, and with phosphoglucomutase, again, the phosphorus is, is moved onto a different carbon of the glucose. So remember, phosphoglucomutase can move in either direction, either converting G1P to G6P or G6P to G1P. I'm just trying to think if I missed anything here. Okay, so the first step of debranching is opposite of what the branching enzyme did in glycogenesis. Go back for a second. Okay, and then we have our last reaction. So we have G6P from our last reaction, and then we have G6P phosphatase, which is going to remove this phosphorus or phosphate group from the glucose molecule, and we're left with plain old glucose. So in this way, we've taken the glucose from the glycogen molecule and made it pure, free glucose. Now, this reaction can occur in the liver, kidney, and intestine and allows the liver to generate free glucose for maintaining blood glucose levels. Okay, so all those reactions can occur to a glycogen molecule in the liver to free glucose into the blood to maintain circulating levels. However, if we'll remember, we do not have G6 phosphatase in the skeletal muscle. Therefore, glycogenolysis in the muscle doesn't impact serum glucose levels because it can't release free glucose. Instead, we have G6P that stays in the muscle and is oxidized via the TCA cycle for the energy needs of the muscle itself. So any muscle glucose released from muscle glycogen is oxidized in the glycolytic pathway in the muscle. Okay, so here's the graphic that I was talking about earlier that demonstrates how the debranching enzyme works. So here we have our glycogen molecule with our linear glucose polymers and our branches on it, okay? So then we have the transferase activity, which takes the last three glucose molecules on the non-reducing end of the branch and moves them onto the linear glucose polymer. So it's making this linear glucose polymer longer using the 1,4 bonds. And then we just have one glucose left on this branch. And then we have the glucosidase, 1,6-glucosidase enzyme activity, I should say, which then releases this last glucose in the form of free glucose. So, and then you can see the result is that we have one less branch on the glycogen molecule. Okay, so this is just an overview of glycogenolysis that we just talked about. So we have our glycogen, then we have glycogen phosphorylase that produces G1P. Okay, so it releases the last glucose from the non-reducing end in the form of G1P, and then we're left with a glycogen molecule with just one less glucose on it. Okay, what I should have drawn here was the addition of a phosphate from ATP and converting it to ADP. So the phosphate from ATP is added onto the glucose. We have G1P, and then we have the enzyme phosphoglucomutase, which moves the phosphorus or phosphate group on the glucose. If we're in the muscle, this needs to be utilized for oxidized to be utilized for energy. However, in the liver, we have G6 phosphatase, which can then remove the phosphate group from the G6P and allows us to have free glucose. Okay, so we know that glycogen phosphorylase is our main enzyme that's used to release glucose from the glycogen molecule. So let's talk about regulation of glycogen phosphorylase. Now, this might get a little bit confusing because it's kind of opposite of what we talked about for regulation of glycogen synthase. Okay, so just like glycogen synthase, glycogen phosphorylase has two forms. A is the active form, B is the inactive form. Okay, so let me think if I can say this and keep it straight. Okay, so just like we had when we were talking about glycogen synthase, we had our kinases that added on our phosphate to inactivate glycogen synthase. Here, one of our major enzymes is a phosphatase, which takes off a phosphate molecule. Okay, so intercellular CAMP is increased in skeletal muscles in response to epinephrine. So epinephrine is the first messenger. It activates CAMP, which is the second messenger, which activates phosphorylase kinase. Okay, so phosphorylase kinase phosphorylates glycogen phosphorylase B to convert it to phosphorylase A, which begins glycogen breakdown. Okay, so this is kind of the opposite of what we're talking about with glycogen synthase. So for glycogen phosphorylase, our active form has the phosphate group on it. Okay, our inactive form is lacking the phosphate group. Okay, so if we have phosphorylase A is the active form is converted to phosphorylase B with the removal of a phosphate via a phosphorylase A phosphatase enzyme. Okay, so let's return to this graphic that we had before, okay? So we talked about this top form, so we have these kind of stress hormones, if you will, that are saying there's not enough glucose, okay? And we talked about how when that happens, protein kinase A is activated and then stops glycogen from growing, okay? So we have the same reaction that's occurring, okay? The same hormones that are involved. We have our first messengers, we have that they impact our second messenger, and then we have activation of protein kinase A. However, protein kinase A, which adds a phosphate group and deactivates glycogen synthase, actually activates phosphorylase kinase, okay? And that in turn adds a phosphorus group onto the inactive form of phosphorylase and makes it active. So this really makes sense. So if we have these hormones that are saying there's not enough glucose, we want to stop the growing of the glycogen molecule or the storing of glucose, and we actually want to activate the breakdown of glycogen so that we have more free glucose. Okay, so we have so they have the opposite impact on our regulating enzymes. These hormones inactivate glycogen synthase and activate glycogen phosphorylase. Okay, so synthesis and degradation of glycogen is regulated by the same hormonal signals. So elevated insulin results in overall increase in glycogen synthesis, and elevated glucagon or epinephrine in the muscle results in increased glycogen degradation. And this really makes sense. So glucagon acts on the liver only, whereas epinephrine acts on the liver and muscles. So the action of phosphatases always opposes that of kinases, okay? So kinases add a phosphate group on, phosphatases take the phosphate group off. And depending on which enzyme it is, depends on if it's activated in its phosphorylated form or inactivated in its phosphorylated form. Okay, so lack of enzymes that we talked about can cause, the lack of enzymes that we talked about for breaking down glycogen can actually cause the buildup of glycogen in various parts of the body. So when we can't break down glycogen, these glycogen molecules can just become enormously large. And the inability to degrade them can make the cells engorged because the glycogen molecules are so large. So when we can't break down glycogen, also, not only do they become large and provide this kind of, I should say, maybe like a physical issue, because they're so large, they decrease the function of the cells, but they also make it so that we can't break off glucose from the glycogen and utilize it as an energy source when we need it, and also use it to increase our blood glucose when we need it. So the diseases that have these lack of enzymes are pretty rare, but they are also pretty dramatic in their consequences. And the extent of the debilitation from these diseases depends on the severity of the mutation causing the deficiency. Okay, so I'm going to go over some of the more common types of glycogen storage diseases. So keep in mind, these are all quite rare. So the first type is type 1 von Gerich, I think I'm saying it right, Gerich's disease. So this is the most common type of all glycogen storage diseases, and occurs when there is a deficiency of G6 phosphatase, resulting in the inability to break down glycogen and therefore the accumulation of glycogen in the liver and kidney. And when we have this growing of these glycogen molecules in the liver and kidneys, they become larger, okay? So the symptoms of this type 1 von Gerich's disease are growth retardation, okay, which makes a lot of sense because we're not able to utilize glucose when we need it by breaking down glycogen, and also lipolysis and ketoacidosis. And that's because we don't have enough glucose in our circulating blood glucose because we can't break down liver glycogen. Now, it is possible to survive with this disease if someone is eating small meals with glucose frequently, so in order to kind of maintain blood glucose levels. And if high protein diets are avoided because they can cause excess gluconeogenesis, and that causes more like glucose storage in the form of glycogen, therefore growing these molecules that their body isn't able to break down. The next type is Pompe's disease, and I've seen a child with this, and what this disease is a consequence of is a defect in the alpha glucosidase 1,4, which hydrolyzes the alpha 1,4 linkages in glycogen. So as you can imagine, we're not able to release the glucoses from the glycogen molecule again. So this can occur uh, have an infantile childhood or adult form. The infantile form is the most common and most severe form, and usually results in death by cardiorespiratory failure. It results in extensive damage to muscle fibers and an enlargement of the heart muscle because the body isn't able to break down glycogen molecules, and also results in excessive accumulation of liver glycogen. And you can see in this graphic here, this baby just has no muscle tone. For those of you who have children that go to their first appointments, their first checkups, they'll often do this test of pulling the baby up just to see what is their muscle tone, and they're really examining them for one of these glycogen storage diseases. Okay, there's also a childhood form of Pompe's disease, which progresses slower but is usually fatal in younger adulthood, and the consequences are mental retardation and loss of muscle tone. And then there's an adult form, which is much more mild and usually not fatal, but does result in muscle weakness. Okay, our next type of glycogen storage disease is type 3, which is called Cori or Forbes disease. And in this glycogen storage disease, there's a deficiency of the debranching enzyme. So we're not able to remove the branches from our glycogen molecule. So once again, we have hepatomegaly because the liver grows because there's so much glycogen stored. We have low blood glucose because our bodies aren't able to basically cleave the glucose from the glycogen storage molecules. We have glycogen accumulation in the heart muscle, in the liver, and in other cells, and it results in growth retardation. But there is normal mental development in this form because enough glucose is available to feed the brain. So the only thing that this disease is lacking is the ability to remove the branches, but it can still take off the glucoses from the glycogen at those 1,4 linkages. So this form isn't fatal. Okay, then there's type 5, which is McArdle's disease, which is a deficiency of muscle phosphorylase, resulting in large amounts of glycogen in the skeletal muscle. So the muscles are not able to use glycogen and therefore must rely on fatty acids, amino acids, and blood glucose for nutrition. And this, again, is not a fatal type of glycogen storage disease. However, patients with this disease are unable to perform heavy exercise, but otherwise they can survive without much difficulty. Okay, and our last form that we're going to talk about is Hare's disease or Hare's disease. I'm not sure exactly how it's pronounced, but it's similar to the type 5, except for the issue is with the liver phosphorylase instead of the muscle phosphorylase. So liver glycogen does increase and reduced growth, and hypoglycemia may be seen because the liver isn't as able to buffer low blood glucose. However, again, if we feed frequently in order to maintain blood glucose levels, we can treat this condition. Okay, so here are the practice questions for this section, and I'm going to end here, and I'll pick up on the next lecture with the next topic.